Okay, so let's start the third session of the course, which is called Introduction to Main Workflows in Mass Spectrometry. And in this session, what I would like is to introduce you on how to operate the mass analyzers that we have seen in the previous session to produce actually workflows that allow us to acquire samples and get the information that we want to get. Okay, so the first concept that I would like to introduce in this session is the concepts of top-down versus bottom-up proteomics. And the difference between these two ones are that in the so-called top-down proteomics, we get tissues, we extract proteins, and then we work with these proteins, for example, for a protein which we have purified from here, and what we analyze are intact proteins. Okay? In contrast, in bottom-up, we again start with tissues or cells. We do the protein extraction, and then to try to simplify the analytical problem, we homogenize this, or we digest these proteins into peptides, which are molecules that are more homogeneous between them than different proteins, right? So somehow we try to control the variability of the analytes that we want to measure, right? So in this case, we analyze intact proteins in top-down. Here we analyze peptides in bottom-up. So in top-down proteomics, we have these tissues, proteins, and we purify these proteins, as we already know. So these proteins, we put them as in droplets in the electrospray, charge. These droplets, the solvent is getting evaporated, then the charges are retained in the protein. The protein is restructured so that the charges are accommodated in a comfortable way, and this goes into the mass spectrometry, into the mass spectrometer. And this is the type of signal or mass spectrum that we get out of one single purified protein. Okay, so you can see that the mass ranges from 500 to over 1,000, different intensities, and just one single protein gives us all these signals. And what are all these signals? So these signals are different charge states. So for example, here you see 37 charges, 38, 39, 40. And this is because here what we are seeing is not the mass of the protein, but the mass divided the charge. Therefore, if this protein weighs 30,000 daltons or 30 kilodaltons, it can have one charge, two charges, three charges, four charges, and so on and so forth. And for every charge state, it will give a peak here. And each peak, of course, so these are not isotopes, as we have seen. These are just mass charge states. And if we zoom in each of these peaks has actually an isotopic distribution. And this is the main challenge for top-down proteomics, because imagine if instead of one single protein that we purify from a cellular extract, we want to measure the whole proteome of a cell without digesting. So this, we would get each of these for each protein. And signals will start overlapping and would be very difficult to understand which signal comes from which protein, right? Even though there are many groups that are working in top-down proteomics that are actually succeeding in actually analyzing not only single proteins, like in this case, but actually mixtures of proteins. And this field is advanced quite significantly in the recent years. Anyway, also what one can do is take, for example, this charge state, which is the most abundant. Okay, in a quadrupole, using a quadrupole, we can isolate this charge state, so we can filter, so we get rid of this, and then we can fragment this and analyze the fragments of this protein. And having done this, so now it's not so important, but these are fragmentation patterns of this protein for a particular charge state, and then we can get information about the sequence of this protein, and also information about potential modifications of this protein. And one nice thing of top-down proteomics is that actually so given a protein, one protein, which would be this one here, the green one, this protein in the cell can be modified in different ways. right? But, for example, one molecule can have a phosphorylation and acetylation, but another molecule in another part of the cell can have the same acetylation and another phosphorylation, or two phosphorylations. So not all the proteins of a given or not all the molecules of a given protein have to be modified in the same way in the cell. And top-down proteomics allow us to identify that this modification goes with this one, and that this one goes with this one, and that this one goes with this one, because we analyze it at the protein level. So we have the connectivity, right? In bottom-up proteomics, as I said, what we have is this same extract of proteins, and we digest and we analyze these peptides. And then, so here would be the same, but in a different way. So we have a mix of proteins. We digest them into peptides. We analyze them into the mass spectrometer. We will see how we obtain all this mass spectrometry information later. But the question, once we have this, the question we have is which peptides were in my initial sample, right? And from here, from this information, we can deduce which peptides we have in the sample. Then we can ask, given these peptides, which proteins I had in my sample. So we deconstruct and we have to construct again. And if you see in the slide, there is one extra protein in the results, which actually was not present in our sample. And this is because while doing this, we can have errors in identification. And these errors in peptide identification propagate at the protein level. And then we can have false positives. So we can say, well, this spectrum here, it was from this peptide, but actually we assign it to a wrong peptide, right? And then this peptide was from this protein that is a false positive. So we will see during this course, especially on Thursday, how important is to control error rates in assigning peptides to spectra, and how important is to control that this error does not propagate neither at the peptide level nor at the protein level.
Sorry, so in the top-down technique, you don't digest the protein before loading?
Exactly, we don't digest the protein in the top-down technique.
You are not limited about the molecular weight? For example, if the protein is too big?
So we are not limited because, oops, sorry, now I don't know where am I. Okay, so we are not limited because of the charge states. Because for example, this would be 30 kilodalton, but actually what we are measuring is in the 700 mass over charge rate because of this. Of course, the bigger the molecules are, the less efficient is the ionization and the ion transmission into the mass spec. And therefore, we have less sensitivity and basically less sensitivity, and because of this ionization problem.
What you're seeing in this picture is the whole protein without fragmentation.
What we are seeing here is the whole protein without fragmentation. Okay?
But the molecular weight of the protein, can you know, or is it difficult to know with the multiple charge?
Well, so what I would do is, so as I know that this is m over z, so I can multiply this number by 41. And this gives me the approximate molecular weight of the protein. Right? Any other questions? Okay. Right. Then what we get in this, so we come back to the bottom up. Okay. So we come back to the bottom up approach where we analyze peptides. And in this case, what we have is, as we have digested the proteins, what we lose connectivity. So what we identify is this peptide with this modification, this other peptide with this other modification, and this third peptide with these two modifications. But actually, we don't know whether this goes, whether in the same protein molecule, these are present together or not. Okay, because we don't know. Yes, we lost connectivity. Is this clear? I didn't explain it very well, but it's very, but it's very important. It's clear, Right? I mean, this is important, for example, for histones, where this the histone code, and you want to see whether the acetylation at lysine 4 is present at the same time that in lysine 27. So if we analyze peptides and not proteins, then we actually don't know whether they are present in the exact same protein molecule. Okay? We know that both are present in my sample, but actually whether they come together in the same molecule, this we lose with the bottom-up approach.
You can maybe cut different, for example, you have also the same pieces of the protein, always the same, because you use the same enzyme. Right?
Exactly. I mean, we could do other approaches, like changing enzyme, to actually try to get, for example, here I got these together. I could use another digestion enzyme to try to get these two together. This is a good approach to actually recover connectivity.
Maybe it's the question. But if you do the top down, you analyze the whole protein. So is the accuracy is higher than the bottom up because the whole peptide pool has to fit one protein. So you identify the peptides.
So can you repeat the question?
So one peptide may belong to different proteins, the sequence.
Yes.
So it's from the bottom up when you analyze the peptide. So if you analyze the whole protein, so all peptide patterns has to fit to one protein. So the accuracy is higher. So because the protein is the protein you identify.
Yeah, well, so we will also, tomorrow we have a dedicated session on quantitation, but important thing is that peptides, as you said, can be shared between different proteins. I mean, we all know that there are some domains that are very frequent in several proteins, and this can have shared peptides. So for quantitation, for instance, we can use everything when we do top-down in a purified protein. When we do bottom-up approaches, we have to use peptides that are unique for that particular protein. Okay, and we will see this in the quantitation approach. Okay. Okay, so then let's move to how we operate mass spectrometers, right? So we are already familiar with the mass spectrometry schema of ion source, analyzer, and detector. We have seen also several mass analyzers, and we know that we can operate them as a screening mode, like the Tamoy flight, ion trap, and the two based on Fourier transform, like ICR and Orbitrap, and some others that we can apply them as filtering devices, right? Like the quadrupole and the ion trap. Okay, and then, so depending on how we operate our mass analyzers, we can have one information or another. So for example, let's say, so I have a chromatographic peptide separation, so these are peptides, and I have a mass analyzer, and I operate it in a scanning mode, right? So what I will get is something like this, right? So what we call a mass spectrum. In this case, we could call it MS1. We will see why afterwards. And what it tells me is that in this mixture, there is the red, the blue, and the green compound, and that they have different mass to charge ratios and different intensities. If I zoom in each of these peaks, actually this is not a single peak, but as we already know from the isotope pattern, it's a complex peak representing all the isotopes. But then I can also operate this simple mass spectrometer that has only one mass analyzer, one ion source, and one detector in filtering mode. Okay, and I can say, well, there are several things eluting from my column because the sample is exactly the same, but now I'm interested in that single peptide, which is the red one. Therefore, I set up the voltages here so that only the red one can pass through the mass analyzer and hit the detector. And what I will see is something like, well, if I record this on time, I can monitor this signal. I can see that something is appearing at this time point, and then something else is appearing in this other time point. If I just get a snapshot of this, not a time course, but a snapshot, what I would see is just one single peak with their isotopic distribution and in a certain mass range, but nothing else in the mass spectrum, because I'm only filtering for that particular target. So what I showed before in the last session is that current mass spectrometers are not such simple as just one mass analyzer, but they are combining different mass analyzers. And normally, so this would be one of the most common setups. in mass spectrometers that have one first mass analyzer followed by a fragmentation cell that allows us to actually fragment our molecules and get information about the structure of this molecule, and a second mass analyzer. And conceptually, we could operate each of these mass analyzers either as a scanning or as filtering mode, right? And different combinations will allow us to have different patterns of acquisition. So the first MS acquisition mode that I want to discuss in this course is what we call the discovery or screening proteomics, and also called top-N methods or shotgun proteomics that you have probably listened or heard about, and it's to just get or answer the question of what proteins are in my sample. Okay? And here, what I would start by doing a screening mode in my first mass analyzer, right? So there is the second, but I don't draw it here because I'm not using it. And the first question is, what is going out from the column? So what masses are in my sample? Okay, and then I get my MS1 here. Then I say, well, I know that 3 masses are coming out of my sample, but what are they? Which sequence do this peptide have? Okay, and for this I have to fragment them and get information about their structure or their sequence. And what I do is then I change, and instead of using the first mass analyzer in a screening mode, as I have used initially, now I set it up in filtering mode and say, well, first let's go for this red one. And why this red one and not the other? Well, just because it seems to be the most intense here. So I can say, start with the most intense, so I filter, so I isolate this one, right? So the red molecules here, I fragment it, and then I generate a lot of fragments derived from this that gives me what is called the Ms2 spectra. The Ms2 spectra is not much different from the Ms1, but it's just that this contains only information about fragments of a particular molecule. Okay, so here I isolated the red one, I fragment it, and I record which fragments do show up from this particular molecule. Okay, and then I get sequence information of this red one. Then I go for the second one, and so I take the green one, I filter in the first quadruple or mass analyzer, I fragment it, and I analyze all the fragments, and I record them as a second Ms2 spectra, right? And this gives me information about the identity of this molecule that was showing up in here. And then I do exactly the same for the third molecule, which is the blue one, and I get also my Ms2 spectra, right? So in MS1, in this type of acquisition, I'm using only one mass analyzer first in a screening mode to know what's going out from the column, and then I combine both mass analyzers, one in filtering mode and the other one in the screening mode, to actually know what exactly is the sequence of each of these 3 peptides. Okay? Then I can repeat this several times, right? So the structure would be 1 MS1, then I do a top N, in this case would be a top 3, right? Because top 3 means that I'm selecting the 3 most intense ions to be fragmented and to get sequence information. This could be top 5, top 20, top whatever, right? But in this particular schema, it's top 3. So I get a picture of what is the eluting from the column, and then I select the first, second, and third most intense to get sequence information. And then I do exactly the same here, right? So I repeat the cycle. So I start with another MS1, and now what I see is that instead of these 3 things, other things start eluting. And then I would pick other things here and to do the MS2, so the following MS2 to get the identity or the sequence of these peptides. Questions here?
I understand.
You didn't understand? Okay, so here I'm taking a snapshot of what's going out of my column, right? And I see that there are 3 compounds with different masses and different intensities. But then the next question that I have is, well, what are they? And then, so I have to isolate them. So first I isolate the most intense, I fragment, and I get fragment information. We will see this after, how to interpret this spectrum. But at the moment, what you need to know is that with this fragment pattern, we can identify the sequence of the peptide. So this gives me information of what is this peptide. This one gives me information of what is the green peptide, and the blue spectra gives me information of what is this one. Okay, now I'm done with this, so I take another snapshot. And now what I see is that, well, the red one is still there. It's still eluting from the column. Maybe it's a very abundant peptide, but other things start also appearing. And now I repeat exactly. So I want also to select, for example, now I know the red one I already know, so I don't want to sequence it again. But for example, this yellow one, I have no information before, so probably I want to sequence this, also the brown one, and maybe also this one here, right? So I would select these ones for further fragmentation and to know, yes, Geoffrey.
And in terms of quantification, the red one should be resequenced again every cycle, if you see.
No, and we will see now. We'll see now why. Okay, so I will answer you in a couple of slides. So the thing is that what I'm getting is different cycles, as you can see here. So 1ms1 followed by, for example, if I'm only sequencing the 3 most intense ions always, so I have 1ms1, 3ms2, 1ms1, 3ms2, 1ms1, 3ms2. So the ms2 actually give me, again, information about what is eluting from the column, right? What is the exact sequence. And the ms1 allows me to decide what I have to sequence, because otherwise I don't know what is eluting from the column. Okay, and this is what we call in black. This black is the cycle time. So this is a cycle from one ms1 to the second to the following ms1. This is a cycle, and we repeat these cycles over and over. And then coming to Geoffrey's question, so how do I quantify this, right? So we know now that the ms2 gives us information about the ms, what is eluting, right? But what I can do is actually say, well, now I know what the red one is. Okay, fine. Let's put all the ms1 together. So I just for visualization purpose, and then I follow the red one, and I see that it was very intense, less intense, and disappearing. For example, now we follow how it was eluting from the column, the yellow one. So it appears here, it goes up, down, down. So I can do these chromatographic profiles in time, and then I can use area under the curve to quantify. So I use ms1 for quantitation, ms2 for identification. Okay? And with this strategy, which is one of the most common strategies for identifying and quantifying proteins in complex systems, and with current mass spectrometers, so we can, for example, with the Orbitrap Fusion Lumos, we can go up in yeast with no fractionation over 3,000 proteins quantified, and human also similar results. And with fractionation, we can reach around 4,000 proteins, which is the amount of proteins that from the 6,000 open reading frames that yeast have. So it's the amount of proteins that are thought to be expressed in a given moment, right? Similarly to humans, right? So human has around 20,000 open reading frames, but in a given cell line, in a given moment, it's thought that no more than 10,000 proteins are expressed in the cell. And this is all the information that we can get with the latest generation instruments, right? So with this instrumentation, what we can do is we can identify with the ms2 and quantify with the ms1 all these peptides. Well, all this. So what we do is we quantify and identify peptides, and then we translate it into proteins. So questions?
Would you say fractionation is online fractionation with your HPLC, or?
So this can be online fractionation or offline fractionation, like size exclusion or a gel, and we cut several pieces, whatever.
And all this information you obtain manually? So you are selecting speech and then you perform the second analysis, or the spectrometer analyze everything and after that you get the information? I mean, for each peak, you say after that analyze this peak and then you obtain, or
so
everything is doing at the same time?
So how we do it is we set the the mass spectrometer in a way that we say, well, whatever comes out here, please select the 3 most intense and for fragmentation, and then it fragments. And then in the second one, so it would be the same, but what we do is we say to the mass spectrometer is take the 3 most intense one, unless it has been sequenced before. Okay? And then we are progressing through the whole sample.
Sorry, but the Ms1 is a protein or a peptide that is fragmented in small peptides?
So here in Ms1, so we are in this case we are always working with peptides. Okay, so I introduced, this might not be clear, I introduced before top-down and shotgun proteomics. We will be working during the whole course mainly with bottom-up proteomics. Okay, so always analyzing peptides that are digested from a protein sample that have been digested, and then we analyze the peptides. There.
There's, for instance, one protein which never been in the top 3, but it's always top 4. We'll never sequence that protein.
Yeah. So this is true. So what current mass spectrometers allow us is to go to top 20 to top 30. And not only this, but with this trick that saying, well, if it has not, so sequence the top 3 unless it has been appeared before. We can go actually quite deep in quantitation and identification. Okay, good. So another thing, So another thing that we do normally is we have these chromatograms, and then we can separate them in 2 dimensions. So here we have time, but actually we know that each of these corresponds to a different mass to charge ratio, right? And then we can introduce this mass to charge ratio, so this same picture, we can actually visualize it like this, okay? And then it's quite easy to flip it over, right? And get this visualization. And this is something that actually we call 2D maps, and we like a lot to use them or visualize data like this. And what we can do, for example, or what happens with these screening methods is that when we have samples, because of what Geoffrey commented now, or also because of these sampling effects that we discussed before, is that if I have 4 biological replicates, so it can happen that at the MS1 level I see everything, right? But at the MS2 level, so what are the things that are identified, which are colored here, whereas the gray ones are not identified. So I can see that the identification here, it's these 3, whereas in the second sample, even though it's the same, so I identified some other things, right? And this brings problems or challenges into protein identification and quantitation. But actually, we are quite good at saying, well, you know, so I didn't identify this spot here that has this retention time and this mass, but I intelligent enough to say, well, this is quite likely to be the red one, right? And what we can do, and what we are doing, is actually sharing information between runs and sharing this identification. So even though this feature here, or this peptide here, has not been identified in this sample, if I have replicates, I can say, well, this feature probably is this one. Okay? This is very easy to do manually, right? But given the amount of analytes, we cannot do manually, and we have to do it computationally. And computationally is not when you are dealing with, instead of 5 analytes like here, where you are dealing with thousands of analytes, is also not so easy to implement, and that computers can do it in a proper way. So in this case, so this is something that we can do. We normally do in many workflows, but depending on the sample, it's not also, the best solution. Okay.
Do you have different samples, or the same sample but different replicates?
Well, so this I made up, meaning that I did the slide, but this could be the both things, either biological replicates or technical replicates. And this sampling effect, you would see in both cases. Okay, so no matter whether they are technical or biological replicates. Okay, so the advantages of this screening or top-end methods is that actually we have quantitative information of all peptides and proteins, because from the MS1, we can extract these chromatograms, but actually we don't have information of identification for all the things, right? So we have more information at the quantitative level than in the identification level. So not everything that we record, we are able to fragment and identify. And the limitation is that are two main ones. The first one is the chemical noise, so meaning that samples are so complex, we are dealing with 100,000 analytes, that there are a lot of interferences. So meaning that there are a lot of peptides that weight slightly similar and the signals overlap, right? And this is a problem when we do, or a challenge when we do MS1 quantitation. And the other thing is linear dynamic range, in which, so mass spectrometers, as you know, are very sensitive, so we can measure things at even at the zeptomole level. But the thing is how sensitive we are when this molecule is together with another very abundant molecule. And here is where the limitation comes, and this is what we call the limited dynamic range. And this goes to 4 orders of magnitude. So if you have, for example, in your serum samples, albumin that is very high abundant, anything that is 4 orders of magnitude less abundant than albumin, the mass spectrometer will not be able to see it. And this is also why there are these depletion approaches to get rid of really abundant proteins. Okay?
To do the MS2, do you lose a lot of your sample that you kind of run everything again?
Yeah. So one thing that I didn't comment on is that actually, so each of these cycles is 1 second, more or less. So we are very fast at doing this. And so we can actually reproduce very well the elution profiles. And so in one shot, we get all this information. Okay, let's then move to targeted approaches, right? So tomorrow we will dedicate it, or we will review the workflow that I have shown of top N and screening proteomics. Wednesday we will go into targeted modes, right? And basically we will see in this course 2 targeted approaches, which are SRM and PRM. Okay, and now let's introduce these 2 acquisition modes. So again, so if you have a simple mass analyzer with a filtering mode and a peptide of interest, I can actually monitor this a long time, easy. Right? And this is an old technique that is called selected ion monitoring. So you select an ion or a peptide and you monitor. Okay? This is what we are doing here. Here, actually you have information about the mass of this peptide because you are filtering, but you don't have any information about the sequence of this peptide. And it can happen that 2 peptides weigh the same as, for example, here you can see that there are 2 things that are eluting at different times but have the same weight. So what we would like to do is actually, for example, do MS2s. So we can monitor this, we can set a threshold and say, well, whenever this signal is over this threshold, then very fast, let's operate the mass spec in another way, right? So we still filter in the MS1, but then we take this, and instead of sending these molecules to the detector, we send them to the collision cell, we fragment it, and in the second mass analyzer, we set it in scanning mode, and then we get the MS2. So for each of these peaks, we get an associated MS2. So we can monitor a certain peptide, and we can also have information of this particular analyte with the MS2.
So when you talk about a cycle, you mean maybe there's one introduction of the sample in the spectrometry of masses, but then you can do more than one cycle with the same sample, with the same electrospray or not? Or it means another electrospray. You understand what I mean?
Yeah. Mm-hmm. Cycles is when we repeat something. So in the screening proteomics approach that we saw before, it was from one MS1, then I do several MS2, then I start another MS1. So this would be a cycle. And this...
If example is inside, you can do different cycles with the same introduction of the sample.
Yeah, so the sample, so this is a dynamic process. So the sample is continuously getting into the mass spec. So I have to do things very fast because otherwise sample is not there anymore. Right? So what I try is, for example, in here, so I'm monitoring things, and while this is reaching the detector, very fast I have to stop this acquisition mode. I have to switch it to this, to get this, and go back to this. Right? But this has to be very fast, otherwise I will lose all the signal. And actually mass spectrometers now are fast enough, so we can do this in milliseconds. Right? So in 50 milliseconds, 100 milliseconds, we can actually change from this acquisition mode to this one. Whereas the chromatographic profile, so this is normally in the 20 or 30 seconds range. So we have plenty of time to actually change the operation mode and back again to the monitoring.
But the monitoring would be to be able to quantify.
Exactly, yes.
So the second one is to identify the...
So here we quantify, here we identify in this case. Yeah? Okay, nice thing also in mass spectrometry is that actually to reproduce these elution profiles, I don't need to monitor all the time, right? So I can, for example, monitor every second, and I will be able to actually join the points and do this elution profile, right? And this allows me that between one acquisition and the other, so in this second that I have, I can do other things. For example, triggering an Ms2 as we showed before. But also what I can do is say, well, you know, instead of monitoring this peptide, I want to monitor two peptides, right? Oops. And so in between these 2 points, I can operate the mass spec in another way so that I can multiplex. So in this way, I can monitor several peptides in one single run. And this I can do for example, 10, 20, 30 peptides that corresponds to proteins. And it's a way to multiplex and do, the equivalent would be, for example, to do 30 Western blots on that particular sample, right? And this is because I have time from one acquisition to another to do other things. And I can actually do this, I can monitor this, I can get the Ms2, and in the meanwhile, I can also do another peptide and get its Ms2. So questions, because from here on it gets complicated.
So are these the inclusion lists?
No,
No, it's another thing.
it's another, totally another thing.
Environmental.
What?
Environmental.
So this is similar to an MRM or SRM targeted experiment, right? The difference, and we will, because this is the introduction to actually SRM. And here is that I'm only monitoring the mass of my peptide of interest. And then from time to time, I decide that, well, you know, something is eluting, so I also want to get an Ms2. Okay? And these questions allow me to go to the next slide, which is selected reaction monitoring, which is the next step on targeted proteomics.
And then the process, here you have these two bits overlap each other, then you can multiplex.
Yes, yeah, I can. So even if they would be overlapping, I can multiplex because here, so I don't need to monitor it constantly. So I only monitor it, so I only see whether it's there or not once every second. So I have a whole second in between one measurement and the second to do whatever I want. And I can use this second to actually monitor another one. Okay, so selected reaction monitoring is the same, but with two mass analyzers. So here, what I do is I filter. So as you can see, in both cases are in filtering mode. Okay, so I have a peptide of interest. I monitor it. Well, so I let it in. I fragment it, and then in the second mass analyzer, now from all the fragments that this peptide generates, I only monitor one of them. And what this allows me, so this double filtering allows me to get rid of other interferences, because actually the probability that two peptides that have two peptide fragments here that have the same mass, so that both were able to pass this filter, actually came from the same, or peptides that have exactly the same mass is very unlikely, right? Is this clear? Right? And so this double filtering allows me to get better signal to noise ratio and therefore much more sensitivity. And SRM is currently the most sensitive way to quantify specific proteins in specific samples, right? So this double filtering. And again, similar to what we have seen before with just not the fragments, but with just the peptide, what we can do is whenever this intensity goes up to a certain threshold, I can say, well, you know, let's get an Ms2 here, an Ms2 here, so that I have also identification here, right? And in these cases, what I do is I stop for a moment recording this, and I change the second mass analyzer to a screening mode, right? And this allows me to actually get all the fragments of Ms2. Okay, so here I'm doing this. I'm monitoring both the peptide and one of its fragments. It's going up, down, and whenever it's going up to a threshold, I say, well, now I operate the mass spec in a different way. So I fragment it and I screen, not filter, but screen all the ions, so I get information of the sequence. And I can know this peak corresponded to peptide A and this peak corresponded to peptide B. Okay, and similar to what we have seen before, we can actually, we don't need to monitor all the peptide and the fragment all the time. So I can monitor them once every second. And this allows me to do other things in between again, right? And what are these other things? Well, for example, I can monitor a second fragment of this given peptide. And what allows me this is to actually see that if this is my peptide, right, of interest, so the fragments, both fragments should coelute, okay, because the peptide is eluting at one signal at one given moment. And therefore, what this allows me to say, well, this was the signal because it coelutes with a second fragment of my peptide, not this one. Okay, and then, so if I do this for several fragments sequentially, I can get information also about the sequence of my peptide without having to do the Ms2, right? And other things that I can do is again multiplex. So I can acquire not only peptide A or the red one, but also several fragments of a second peptide. So in general, what this allows us is that we have identification and quantification in once, right? So because if we monitor several fragments, I can actually have area under the curve of this for quantitation, but the fact that several fragments are coeluting gives me the identity of this peptide. So with SRM, I have both identification and quantification in one experiment. I have improved dynamic range because as I'm filtering, so this little or low amount molecule is not competing with albumin because albumin, I'm getting rid of it during the filtering process both in the first and the second. Then, therefore, I gain improved signal to noise ratio. I gain also sensitivity. And as we have seen, I can multiplex and measure more than one peptide per protein. Okay? And limitations are that, as in any other targeted approach, I'm only getting quantitative information for the peptide of interest, right? Because anything else is filtered out. Okay?
But you need to know very well what you're looking for in advance.
Exactly, yes. So you know that this transcription factor is key for your process, and then you select unique peptides for this, and you know you have to study a bit the peptide and which fragments it will generate. Yes. So this we will see it and we will do it on Wednesday.
The filtering mode, or the first step, you need a minimum quantity to start all the process. Because if the sample is too complex at the beginning, for example, although express transcription factor, till you have enough protein in the first step, you can do the rest of the analysis. So, I mean, what is the way to know you have enough protein to do all the steps.
Okay, so currently these filtering strategies are more sensitive than the discovery approaches, right? Because of this double filtering and this improved signal-to-noise ratio. But this does not mean that we see everything with these approaches. So there are things that are still very low abundant, and we don't have enough ions to actually get a proper signal-to-noise signal in this, or ratio of our signal. And then we need to, for example, fractionate. Yeah.
So if I get it right, you select a specific peptide sequence for mass analysis, right?
Yes.
So why in the end you can get two quantifications?
Here?
Yeah.
So because unfortunately there are peptides that weight the same, that have the same mass. So for example, given a sequence, any permutation of amino acids, actually the peptide will have the same mass in the first mass analyzer. And also, unfortunately, there are things that have the same weight, so two peptides that have the same weight, and also have fragments that have the same weight. And this is why we get, instead of one single nice peak, we get some interferences.
So it's also possible to get more than two.
Yes. Yep. Okay, so we've seen two main workflows, so the discovery and the targeted, and we will review them tomorrow and Wednesday in more detail. And now we know already how to operate, or we have seen how to operate the first and the second mass analyzer, but I would like to dedicate the last minutes to actually what's happening here in the fragmentation cell. That this is something that we have not talked about at any moment, right? And in here what we do is we isolate these peptides and we try to fragment it to generate sequence information. So this is a peptide, and in these peptide fragmentation strategies, we use them in a way that each molecule only breaks once, right? And it can break at different points, and depending on where it breaks, we give them a different letter. Okay, so for example, if it breaks in between or in the amide bone, within the amide bone, like here, so this fragment here will be the Y fragment, and this will be the B fragment. Okay, and then we have A fragments, B and C, A, B, C, and so on and so forth. And here we have X, Y, Z, right? Because, so from one amino acid to the other, it can only break in 3 different bonds, okay? Depending on the amino acid, it gets the number, right? So letters means in which bond the cleavage occurs or the fragmentation occurs, whereas the number indicates the position of the amino acids. So for example, in ABC, so we start counting amino acids from the N terminal, whereas in XYZ, we start counting amino acids from the C terminal. So these are the Z or XYZ1 ions, XYZ Z2 ions, so on and so forth, right? This is a nomenclature that we will see also on this afternoon, the tutorial on how to sequence these peptides and how to get information out of this, okay? Then, so the first collision or the first fragmentation strategy that I want to discuss is the collision-induced dissociation, or CID, and there are 2 ways of breaking peptides using this strategy. And this is one of the most common strategies to do peptide fragmentation. And the two ways of doing are the resonant excitation, in which we apply radio frequency to excitate the gas, for example, this can be, so these would be our peptides. These black dots would be nitrogen or helium. We excitate them so that they collide into our peptides, and then they fragment it, right? And we also apply a certain radio frequency so that there is not too much energy so that it breaks multiple times, but just enough energy that each molecule breaks at least once. And then we get, with CID, we get Y and B ions, right? And you can see B and Y, and all this. And the other thing, and the other way to apply this fragmentation is actually to pass, or the beam type collision activation is to pass our peptide very fast through a gas chamber, right? So here the peptides are in motion, whereas the nitrogen is static, and then they also collide, and we fragment this, and this gives us normally Y series, so only the Y fragments. Okay,
What's the first type of induction of collision?
Resonant excitation. So what we do here is we excitate the nitrogen particles, and they collide into our peptides.
Excitate like how?
What?
How do you excitate the molecule?
With a radio frequency. Okay, so then what it happens is that we excitate this molecule. So this is not very important. I mean, the mechanism itself is not very important, but the concept is important, right? So that this gets excitated, there is a rearrangement of charges that leads to a break of the molecule into pieces, B and Y. Okay. So one important challenge for this type of peptide fragmentation is when we analyze phosphorylated peptides. And as you know, phosphorylated peptide or phosphoproteomics is one of the main applications and one of the main questions that we want to answer with mass spectrometry-based proteomics. And the thing is that when we use this collision-induced dissociation, what happens is that the phosphate moiety is very labile, and therefore this rearrangement of charges, what induces is that not the fragmentation of the peptide, but actually the loss of the phosphate. And what we get is not fragments of this peptide, but actually the entire peptide without the phosphate. And instead of getting so this would be a fragmentation spectra that we would like to have a lot of peaks with a lot of information because it corresponds to different fragments. What we get is the same peptide, but without one phosphate, without the other phosphate, or without none of the phosphates, right? So we don't get any sequence information out of this. And this is the motivation why other people or some people in the mass spectrometry field started exploring also other fragmentation techniques. And one of them is the second one that I want to highlight today is the electron-based dissociation, right? So CID, it was based in collisions, right? Here, this fragmentation is based on electrons, right? So we put electrons in our peptide mixture in the mass spec, a source of electrons, right? And this, what it does is that this electron makes our molecule unstable. And therefore, again, there is a rearrangement of charges that induces the fragmentation of our peptide. And this generates, instead of B and Y ions, it generates C and Z ions, right? But so this is something that one has to take into account when interpreting Ms2 spectra, but once one knows this is quite simple.
So one point I don't understand, why is only fragmented between the peptide bond and not other parts of the molecule?
So here, so it can be anywhere within the skeleton. It could also happen that it breaks somewhere else, but actually these fragmentation techniques are quite optimized for peptide analysis. For example, it happens, and it has happened, for example, when we analyze other modifications, like for example, adenosyl-based modifications, that actually this molecule or this modification breaks in other parts and then complicates quite a lot the identification. Yes, so but it can.
That to only break
Exactly, but it can happen that if you have some modifications, like for example, the phosphorylation is one of the cases that it does not break in the backbone. Okay, so, and this is the same spectrum that I was showing before, and this is how rich we get, or how rich it can get when we apply another fragmentation technique, right? So this was a phosphorylated peptide, no sequence information with CID or CAD, and actually full of information when we use another fragmentation technique. And then the third and last fragmentation technique that I want to introduce today is the high energy C-trap dissociation, right? And this is a particular technique that is implemented in the orbit traps. So the fragmentation occurs in one part that is called the C-trap, and what it generates is nice. So for normal peptides, it generates a nice series of Y ions. Okay, and this allows us to actually have sequence information or sequence information to identify the peptides of interest. Okay, and with this we would finish this session. So are there any open questions?
So one difference between the collision induced or the electron induced fragmentation is that with one you can break phosphorylated peptides, no, for example, and with the other no. With the electron-based fragmentation.
So the CID, the first one, so for some cases or for many cases with phosphorylated peptides, what it happens is that one loses the phosphorylate, phosphomoiety. Exactly.
So which is the advantage of the first one, of the collision-based compared, I see the advantage of the electron-based fragmentation.
Yeah.
The other has another advantage?
Yes, so that for non-phosphorylated samples, it works much better in terms of identification. Yes.
And is there any fermentation technique that that fragments in the APs?
So the answer is probably yes. Now I cannot name any.
And what happened with the other post-translational modifications?
So
you also use for example, an acetyl group?
So for example, acetyl group would be much more stable than the phosphorylation, right? So this depends on the modification itself. And then for some, the guidelines, let's say, are very clear. For others, there is still no clear solution. There?
On SRM, do we have the database for peptide information? Like you can set up what's the interest peptide, and then you can set up the machine.
Yes.
And what about if you are studying a protease and then you have the fragment not investigated before, so it's a new fragment? I mean, how do you know?
Well, if you have the sequence,
Yes.
we can theoretically know the mass of this peptide and the fragments that it will generate. So we can do everything in silico.
So it can be theoretically calculated, but not experimentally database.
Exactly, yes.
If you have de novo proteomics, for instance, on peptides that are not very well, you don't get the sequence, but you have peptides, would you prefer to use the high collision, or would you still do the CID or CAD to have the monominus information in the line trap? Or is it just, the HCD spectra are so good that you just don't need mono anymore?
I don't have a clear answer. I personally would prefer HCD, right? But
I assume that you don't have time. You have loads of data sets and you just don't have time to fiddle with ammoniums and so you would go for it.
Yes.
You just have a clear why.
Exactly.